Okay, well, my name is Ian Godwin. I'm now the Director for the Centre of Crop Science at QAAFI. I decided to study agricultural science a long time ago, in the 70s when I was at school. And the reason why I did that was because I did a thing on genetics as a school kid. And then I decided I'm going to do genetics, and I came to UQ, a sort of an open day thing. It was called something else then, to work out, okay, how can I do genetics at UQ? And there was this guy who was putting fruit flies into bottles and getting them to mate and produce large numbers of fruit flies, and I thought, yeah, that's pretty exciting, not? And then there was a guy who was doing human genetics, and I thought, yeah, I don't want to do that. And then there was people called plant breeders. And I thought, yeah, that sounds pretty exciting, so I decided to do ag science. And then, of course, I majored in genetics, and I did a PhD in genetics, and then I did a postdoc in the UK in sort of genetic engineering in plants. It was all in plants. That was the stuff that I was interested in. And then I got my PhD supervisor's job back at UQ, and that's my story. That happened in 1990, and I've just been here ever since. But my first international aid and development of a project was on sorghum, and it was with India, and it was to help India develop sorghum transformation. And the traits that they were interested in was insect resistance, because they've got some really serious insect problems in India, and also they wanted to look at well, basically developmental traits, because they've got in India there's 2 growing seasons for many crops. So there's the one that they grow during the rainy season, and then there's the post-rainy season. We were most interested in Australia on the post-rainy season because it goes into terminal drought, which is very similar to the conditions that we tend to grow sorghum in here. And so there was not so much heat tolerance in India because that's going into the cooler season, but here heat tolerance was increasingly of interest, but drought was the big one. So it was quite a large project, and some of the stuff that we did on that project has been applied in India, both at ICRISAT and at our main partner was the National Research Centre for Sorghum, and they started to then use the transformation techniques that we had developed to create a bunch of sorghums with lots of different traits. So that was sort of my first overseas experience. I had projects on taro. The one that we've currently got, well, we've got 2. We have got one on taro at the moment as well, which is also related to climate change. But one that people might be surprised to know is that we're working on a project in the Solomon Islands and PNG on oil palm. And oil palm has the reputation in Southeast Asia of being devastating for the environment, and it is, because people are clearing rainforest. It's a very different situation in the Solomon Islands. All of the current oil palm plantations are grown on places that used to be coconut plantations. Coconut became non-viable as an export industry. Now, although people from an Australian agriculture point of view would think of oil palm as being quite a small industry in the Solomon Islands, it's their #1 export. And the focus of our work has been to look at genetic ways to get better productivity with a mould. It's called white mould or basal stem rot. The fungus is Ganoderma. So it attacks the base of the plant, and the plant actually dies because the xylem vessels get eaten away by this mould. It's a wood-eating mould, and then the trees fall over. That makes them non-productive. We've been taking a genetic approach, but we've also been taking a management approach. So when we started that, so this is a long-term experiment. Our field trial was planted in 2010, and we undertook 2 treatments. One of the treatments was when a normal field is felled, because they usually go for 20 to 25 years, they'll just cut down the trees, put them in a row, leave the trunks there, and then they'll plant new trees. It was pretty obvious that those trunks, especially because there's Ganoderma around, it's a bracket fungus, and you can see these brackets that grow out of the side of the tree. Those trunks are a wonderful source of inoculum. And we thought, we've got to be able to do something about this. So one of our treatments was remove all of those and then cut out the whole base of where the tree was. Now it turns out by about 8 years later, the infection rate of Ganoderma was 4 times higher in the field where we didn't remove the trunks. So being interesting knowledge, next thing is no smallholder has got the money, the manpower, the equipment required to remove these trunks. It's so much easier to actually just leave them rowed up along in between the rows of the new palms. Secondly, some of them don't even do that. They actually just go, and when they decide to replant, they poison the standing trees, and then they grow their new rows between the old rows. So you have these fields where you can look up and see the brackets showering down spores on the new trees, and you know that the infection is going to be terrible. So we thought, well, how are we going to do, what can we do that is going to be an appropriate technology to somehow get it to be environmentally and economically sustainable, especially for smallholders, to take these trunks away. And so we thought, okay, we've got to find a value for this. Now, so the Queensland Department of Ag and Forestry has had a long-term project called Cocoa Wood, and it's about treating coconut palms, because a lot of places were cutting down coconut palms, treating coconut palms to turn that into wood. So we explored that. The oil palm trunk is quite different, so that wasn't going to work. So, you know, we ummed and ahed, and then someone sort of just happened to walk into the office one day and said she was looking at all these projects on biochar and creating biochar as a nursery planting material. Now, so here we are, we're a bunch of geneticists and plant pathologists. This isn't our area, but Jitka Kahanek, who's this young scientist, and Agnieszka Mudge, who's the pathologist working on the project, talked to ACR about what if we can monetize those trunks, turn them into biochar, and then create that sort of circular economy, because when you're getting ready to plant your next lot of trees, you have to grow them up in a nursery. They're usually about 18 months old before you plant them. You don't plant seed, so you plant seed in usually plastic pots or containers, and you have to buy potting mix. What if you can turn the biochar, turn the trunks into biochar, create a potting mix that enables you to not have to import peat, and plus we also know that most parts of, for example, Europe have said we're not going to use peat anymore because it's not really a renewable resource. If we can replace that with biochar, it's a win on an economic sense, it's a win on an environmental sense. So now we've got to the stage where our collaborators have built 2 different types of biochar kits, and they're trialling what the best way is to turn the trunks into biochar, and then that biochar is then going into the nurseries. And I guess I'm using this as an example that once you've been involved in international work for a while, you realise that even though I'm a geneticist, genetics may not be even close to being the best solution. Now, of course, if we do in the long term get genetic resistance to Ganoderma, that's a win-win as well. However, the genetic resistance to Ganoderma is probably only likely to work when the plant is alive. As soon as you cut that down and put it on the field, the Ganoderma is going to be able to multiply on those trunks. So even if we get trees that are totally 100% resistant while they're alive, those resistance mechanisms are likely to be only from a live tree. As soon as you cut it down, Ganoderma is going to grow on it and reinfect your next lot of palms. So that was a pretty exciting one. And then the other one is one that another young scientist from UQ, I don't know if you know Dr. Millicent Smith, she's involved with it, with Brad Campbell. So, and this also goes back in history, probably before either of them were born. I was involved in leading an ACR project. We were collecting taro genetic diversity because there was a disease, taro leaf blight. It was very similar to the famous potato leaf blight when that hit Ireland in particular and created a famine. That same scenario started to occur in Samoa in the early 90s because the Phytophthora that causes taro leaf blight hit Samoa. 95% of Samoa grew one variety of taro. It was highly susceptible. When I say highly susceptible, it meant you could tell when the taro leaf blight had arrived in your field because you'd see these little brown spots on the leaves. Within 2 weeks there were no leaves and therefore no yield. And taro is such a big deal in Samoa that it's actually on their currency. On their currency is taro and bananas because they underpin, they're the traditional staples. So that was quite devastating, but the idea was, okay, if this is going to spread all throughout the whole of the Pacific Islands, we need to collect germplasm now, and then we need to have a mechanism where we can get germplasm into a place like Samoa so that it can be tested for leaf blight resistance. The project itself took 4 or 5 years to get the collection made. We DNA fingerprinted it. We made a core collection. And because there's no real true seed in taro, and certainly if you do collect seed from taro, it's not going to be the variety that you got from the corn. It's outcrossing, it's heterozygous, etc. So they have to keep the collection in tissue culture. And that's a big cost for the Pacific Island countries because every 6 months you've got to subculture them. And then to make sure you don't lose them or lose the genetic integrity of them, every few years you have to plant them out in the field, isolate new tissue cultures and get them back into the tissue culture. That was the happy story. The sad story was we finalised that collection in 2003. The person who led that project was a postdoc called Emma Mace, who now works in QAAFI. Then everyone lived happily ever after, well, except that no one ever has used that taro collection since. Now, partly it's a matter of funding, but partly it's a matter of knowledge that this collection might have something that's useful. Because no one's ever genotyped it. We don't even know whether they're taro leaf blight resistant. And that was what the whole project was about, but we were never able to get the whole collection tested. So FAO have funded the South Pacific Community, Secretary of the Pacific Community, to then subcontract us to look at screening that collection for salinity. And the reason why they want us, why salinity is the low-hanging fruit for them is with rising sea levels, and taro being generally something that's grown in fairly swampy coastal areas all around the South Pacific, and they're the ones that are most likely to get inundation with salt water. So salinity levels are starting to become a bit of an issue there. So Brad Campbell is working on leading that project, but Brad is, like me, a geneticist. He got into it because he's got a DNA fingerprint the entire collection again. And so we asked Millie Smith, as a physiologist, would she help us to develop a salinity tolerance screen? And that work is ongoing. And once we've got that screen nailed, we'll be able to get over to probably Fiji in the first instance and screen that population for salinity. Now it might be there is no salinity tolerance, that's our worst case scenario, or it might be that there are some lines that have got salinity tolerance, but again they mightn't have characteristics that people like. The reason why everyone in Samoa was growing a single line called Niue was because it had the eating quality that everyone liked. And that's not dissimilar to what happened in the 1850s and 60s in Ireland. Everyone liked a particular potato because it was high yielding and gave them the eating quality that they wanted. But that gives you a genetic bottleneck and a susceptibility to when a pathogen comes in. If the population's growing 200 different kinds, the likelihood is there are going to be some that are resistant to whatever that pathogen is. With salinity, we're not so sure, but time will tell. Sorghum in Australia historically was a minor crop. Hybrid breeding made it a little bit more attractive. The development of midge resistance made it even more attractive to farmers because they didn't have to spray insecticides on it. And then as it got hotter and drier, farmers who thought of themselves as wheat farmers who might grow the occasional bit of sorghum on their farm, most of them are now sorghum growers who might grow the occasional bit of wheat or other crops on their farm, because sorghum is the one that stands up to the heat and the drought that climate change has basically delivered to northern Australia in the last 20 years. So sorghum's niche in Australia has been, it's become a larger niche just naturally because of climate change. And so I started working on sorghum in the early 90s. And the thing that attracted me as a geneticist was amazing genetic diversity, amazing plasticity, and also among the cereals. Like we tend, as scientists and also as farmers, to think of cereals are an annual. You plant the seed, it grows up, the seed matures, the plant dies. Sorghum doesn't die. Sorghum's actually a perennial. And that's good and bad, because it does mean that when you want to harvest, from a management point of view, you might have to actually find a way to kill it, which these days is usually a herbicide. But secondly, it means there might be actually opportunities, if it rains at the right times, people can have a return crop. So if you plant a crop of sorghum in September and harvest in January and then it rains, you've got a couple of options that are going to make you more money. One is, well, I'll get a return crop, I'll get a second crop. The other thing is, if you've got beef cattle or your neighbour's got beef cattle, you can say, let's graze it because we've got a lot of biomass here, and which is good cattle feed. So that was sort of the attraction of sorghum. And then within the same species, I mean, it's like the whole cabbage family. You know, you've got Brussels sprouts, cauliflower, all those sorts of things, but it's one species. You've also got forage sorghum, and then you've got sorghum that you can use for silage. You've got sorghum you can use for hay, and it's all the one species. And it's a big biomass crop with, compared to other cereals, smaller, it's more water use efficient, and it's more nitrogen use efficient. So those things in itself make it a wonderful target from the whole, climate change mitigation type of thing. So sorghum has become a Queensland crop because of climate change. But the things that made it, because it's heat tolerant and drought tolerant, it's now getting hotter and it's getting drier. So what can we do? So there's a lot of projects going on in QAAFI that are about heat tolerance, and then we've been doing quite a bit of stuff now with using gene editing with CRISPR-Cas9 to change particular developmental traits to alter the root system, for example. And we've now got sorghums that have a narrow root system where it goes quite deep, and there's sorghums that have a wide root angle, and it's more of a shallow, but you've probably got more root biomass. And depending on the conditions that you're growing sorghum under, they might be beneficial to your particular area. But we think in most of southeast Queensland and even central Queensland, on the deeper clay soils, those deeper roots are going to be the best because they'll give you access to water at depth, and there's often in those heavy clays a lot of stored moisture. In the winter, wheat and barley can't actually access them because their roots don't go deep enough. But you follow that with a sorghum crop, and the sorghum can get to that deeper water, provided there's enough water on the way to get the plant to survive going through. So pests and diseases are major, but let's not forget that many times the range and incursions of pests and disease is a result, an indirect result of climate change. And we can see that in places like sub-Saharan Africa. There are pests and diseases that are going up into the highland areas where they've never been before, and that's because the highland areas aren't as cold as they used to be, and so the pests and diseases can get through their life cycle. If you look at East Africa, for example, all the big cities are built 2,200 to 2,400 metres high. And there's a reason for that. It's because that's a nice place to live, climate-wise, and there's no malaria. Now there's malaria. Malaria has become endemic in cities like Addis Ababa, which was put where it was predominantly to escape from things like that, and it's a nicer climate to live in. Look, I would say the average smallholder that I have dealt with in the Pacific, working on oil palm, climate change they know about, but they care more about the fungal disease, the price of their palm oil, and there's an incursion of a beetle that it's a beetle that prefers to attack coconut palms, but if there's no coconut palms around, it'll go onto oil palm. So that was a very recent incursion, only in the last 3 years in the Solomon Islands. So that's more their pressing interest than climate change. The taro growers are very much focused on climate change, and that's because salinity is affecting their ability to grow and produce taro. But there's also an understanding, I would say, amongst them that they don't feel like there's anything they can do individually to mitigate climate change. It has to be a whole of government approach. And in the Pacific, most of the countries actually point their fingers at Australia and say, you're one of the biggest emitters of greenhouse gases in the world. Can you please stop doing that? And maybe the sea level won't rise as fast. Maybe we'll be able to stop that from happening. Again, a lot of Pacific Islanders would be of the opinion that the cyclones they're getting are more regular and more intense, and most people in the Pacific Island countries would attribute that to climate change. So those are much bigger national type, almost catastrophe type of questions, rather than believing that what they can do on farm as an individual is something that can change. Now it's different in other parts of the world, and probably there are parts of the world where people are more organised and actually also can see opportunities from doing things that do have a positive impact on their footprint, as in make their footprint lower. That's actually going to drive a lot of what agriculture is doing in the tropics and subtropics in the next 20 years. And some of it will be to do not so much with an oil palm grower thinking, how do I change something to make there be less problem with climate change, but more about, if people start closing off their markets to me, I'm going to have to change the way I do things. And it's a powerful argument. And the advice I have, and again, I'm biased because I'm a scientist, is it's great to have a love of science. It's great to be passionate about your science, but the best part of that is to think creatively. How do you use the science that you know and form groups with other scientists to create multidisciplinary teams to solve problems. And I guess I've given you some examples, like the oil palm one. We went into this, we're going to solve the problem as geneticists, and it turns out that it's grown into something much, much bigger than just a genetic response to a disease. You, as a young scientist, look, as any scientist, you can't know everything there is to know, even in your own narrow field. But concentrating on your own narrow field and being open and actively thinking about ways in which to collaborate with people who've got other skill sets, that's how you're going to solve problems. Because these days, very, very few problems are solved by they're not going to be solved by the Albert Einstein. It's going to be Albert Einstein type people who are capable of getting other people around them. A bunch of other Einsteins, well, maybe their name's not Einstein, but basically a bunch of other people who are also passionate about this, who want to go on the same journey, but use their different set of skills to find creative ways to solve problems. And even the stuff, for example, that we're doing on taro. So we need a physiologist to understand things about the salinity and how we test with salinity. But we also need people who can set up the systems. And in other words, it's almost like engineering, setting up the systems to test properly for salinity. And also getting into the smart technology, being able to take images in real time, because you were talking earlier about your inability to get onto your field site because of the rain to take the measurements you needed. If in 5 to 10 years' time the technology is there, you may not have to do that, because it may actually be being collected 24/7, and even at night. Now, those technologies do exist, but they're just too costly and too unreliable. But that was probably the same when cars were invented, 100 years ago. And now, you look at the capability of our cars now, they're driving themselves, they're computers. Where is our ag tech going to take us to enable us to generate the data that gives us the answers we want? I can speculate, but it's almost impossible to imagine. And that's an excitement to me. But you have to have a good scientific underpinning to be able to use and understand those technologies. You need to be able to ask the right questions, test the correct hypotheses, rather than just, let's go measure 20 traits and see and analyse it at the end. You have to have something that you're actually testing. And the other advice I'd give to young scientists, and this is not my own words, it was the former Chief Scientist of Queensland, he said, make lots of friends. Because when you've got a problem, you're going to see a friend to ask them to help. And I thought, that's a really good way to do things. Just know a wide range of people. Sometimes it gives you new ideas. Sometimes they've got a problem and hey, actually, I've got a piece of equipment that can solve that for you. Or I know someone who's got that piece of equipment. So that network is crucial to being a good scientist. It's part of the soft skills type of thing, I guess.